And this is a big picture about the difference, which for this group I think is not useful, so I'll just skip it. And on the ground, my group work in a fashion that start with a seed scientific problem. I'm very uncomfortable proposing general methodologies without seeing it worked out for one example. So my guideline is any new statistical method should at least solve one new problem, or better, or something. And then we generalize. So that's why we're testing it on this data with knockout experiment before we feel comfortable pushing out for other people to use it. And postdoc students are embedded in the lab. They spend 50% of their time in ABL and next to biologists. And I want them to become a little like a biologist. And that's become more possible. I mean, I think Atu's talk this one are kind of going in the same direction, in the sense that John Tukey, who, the fame of fast Fourier transform, Cooley and Tukey. And he was also a statistician who really brought exploratory data analysis to kind of the more respectable platform. And he used to say that statistics play in everybody's backyard. I think now actually bioinformaticians have the opportunity to play in people's front yard too. Because a lot of the data, like the Sioux group, this is new data for them. It's not like they have a lot of experience and good intuition. This is very new technology. The rapid development of technology really drive the need of people like us, and also give us the opportunity to become a little like a biologist, because the data. But you also have to learn what they know and you don't. Oops, this screwed up. Just like the problem I was pointing out to Haiyan. It's the translation problem from different platform. So data collection and pre-processing, you can see it's kind of messy. That's exactly what it is. This line is not nice. I think pre-processing at least 80% of the work. And that's something, again, if you don't do it right, can be a problem. But maybe the stability can help us there. So you have many sources and don't have to worry as much. But it's always a problem. If you have small data set, not enough data, you want to do very well here. This is the BD-GP data. And as I said, Ben Brown, who is a collaborator of Haiyan and Peter for many years, approached me maybe 3 years ago, said, this is a good project for you. And I just actually believed him and got on the project. So Drosophila is a model, like animal, you could call that. And it shares 75% by one estimate of genes with humans. And it's very easy to breed, and it's very fast life cycle, so it has been used a lot. And there's tons of knowledge and trained humans about Drosophila. And this is more urban phrase, brain baby, I think, is to have this high throughput gene expression image. So you start, it's microscopic images, and you have to separate them into different parts, and then look at where they're expressed. And it's 1 terabyte data. I'll call it academic big data. It's not industrial scale big data. And the questions we seek are the following: how many functional regions are there? This is developed biology. The image we're looking at for this talk is about 2 and 3 hours after the embryo start developing. And this is a textbook kind of division of Drosophila for that stage. You can think of this different color like different rooms. When you build a house, you build rooms for different functions. And the organs kind of like that, right? You have to eat, you have skin, you have neuron. So think of this, that's what the embryo tried to do, is try to build these different compartments to carry out different functions. And the genes, you can think of different workers. Some are plumbers and some are painters. And they work together for different teams to build different parts of the floor plan. So we use this as a validation to see, can we use our modern data? This took a lot of work, took decades to come out of this. You do something called laser ablation, so you basically cut and see what happens in development. Okay, so can we use this? So this is the first. And the genes we're looking at for this talk, again, mostly are transcription factors. You know, there's a whole pathway from DNA to phenotype and like texture, morphology. And transcription factors are binding molecules, and they trigger the genes to express. So we kind of exchangeably talk about genes and transcription factors. For this early development stage, there's not a lot of feedback loop, which make us feel more comfortable doing gene network, because it's more cascade and just trigger, things happen. For the data collection, it's using dye chemistry to visualize gene expression patterns. And you have to pick these isolated embryo images. This is high throughput, and Mike Begin's group, also at the IBL, has more high resolution technology, but they can only do like 30 or 40 genes, and we have like 700 genes. So it's relative, and we're trying to use their data to validate part of our results. So this stage, the little embryos are not two-dimensional, right? Three-dimensional. So you have to find the right view to put them together. Otherwise, you really compare oranges with apples. And how do you know which stage? This part we hope to automate later is manually done. Humans, trained biologists, look at them and say, oh, this is the stage, early stage, right? So that's what's happening. It's still very labor intensive. So before we got on the project about 3 years ago, they already did segmentation. And we used Eric Singh's group from CMU's SPEX2 to put them on a standardized ellipse, so we can put them together into a structured data form. And we did more shadow removing. Siqi did a lot of the work. So after all of this, we have now standardized data form. All the images become on this ellipsoid 405 pixels, we call super pixels. The reason we made it very coarse is because computation power, right, for whatever we have. So in theory, we could go back, redo everything with more pixels. Previous work on this data before, because it was already public domain, was more like supervised learning. The humans went in and looked at, oh, this part is called control vocabulary, looks like a gut, so they have label data. And all these people trying to automate that step. We go back to that step, but first, what our main work is like unsupervised learning. If you read the AI literature now, that's the bottleneck, it's unsupervised learning. I mean, I don't know if you follow deep learning. It's a huge amount of like ImageNet, 15 million label data. And that's like really outsourced human intelligence, which was not possible even 10 years ago, right? So you kind of automate that. But without labels, can you learn the structure? So that's what we try to do here. And the spatial information is rich and meaningful. That's basically the foundation for any work based on spatial gene expression. Why? If Nobel Prize is any indication, that the most celebrated gene network for Drosophila is the gap gene network. It's responsible for the division of the embryo into different parts, like that's a human embryo, that's a fruit fly embryo. Not that different, right? And Nuslen Wenhard was awarded a Nobel Prize in the 90s for her work in the 70s. So she figured out, or her group figured out, that these 8 genes are crucial for this segmentation development. It's like a vertical division. And the names are all in German. GT means giant. So this is a gene that the name is very telling. When you knock out this gene, the embryo becomes big or giant. And this is tailless. That means you knock out this gene, the embryo would not have tail, and so on and so forth. But you see that there are 2 giant there. It says that giant depends on where it is. It interacts with different genes. It's just like the plumber does different things in different rooms. So if you clump all the things together, then you're going to lose that information. So if you try to use the whole embryo without the spatial information, you're going to mess up this gap gene network. You cannot reconstruct it. Okay? So I know a lot of people came from far away are falling asleep. I promised Minghua to wake him up. And this, the signature genes in our data, is very coarse because it did 405. So you look at the hunchback. That means you knock out this gene, become a hunchback. And the giant, you visually can put them together, means probably see that they're kind of repressive each other. And they are, because they don't appear at the same time. Okay? So that's how the spatial information can also help you to find out repressive enhancing links. And this is also from a previous, I think it's a book, this compartmentized embryos. They just think there are different patterns, right? Block patterns. And the one question when we finally made our kind of statistical investigation or data investigating precise, can we reproduce these building blocks? Right, that's more precise than, oh, let's just find some interactions, right? That's vague. And we came down next day saying that, can we find these building blocks? And then something rang a bell. Actually, I was talking to my postdoc at the time, Julian Morel, about this problem. And then that's where the idea came up. We were working on sparse coding for computation biology. As you said, I like images. So I've been working with a landslide for 10 years to figure out how the visual pathway works. And if you feed this sparse coding, it's kind of a one-panelized decomposition of image patches from natural images. That's what you come up with. You have this like Gabor wavelet-like filters, basic edge detectors. And these edge detectors, this came from natural images, and the pioneer work of Al Housing, who's at Berkeley Redwood Center, and also Field at Cornell, did this in 1990s. But before that, in the 50s, Hubble and Wiesel did their Nobel Prize winning work later. In V1 cells, they did recording, physiology recording on sleeping cats, and they saw something quite similar. So the cells actually react this with like edge detectors. So you have the two lines of work and produce the same result, because our vision are very much evolved to look at natural images. So there's no surprise, you can kind of take two routes and see something similar. So mostly, this is decomposition, right? You can write now natural images as linear combinations of these little edge detectors. So that's where we started. So we really borrow, that's another part of statistics, is we borrow ideas from other fields. And then we don't have to start from scratch. So you have a head start. We end up using non-negative matrix factorization, which is very similar to sparse coding, but mathematics is different, but the results are very similar. So we put the data matrix into a vector. This is maybe 700 genes. Each has a different gene expression, spatial gene expression signature, into these very coarse pixels, 405. And we don't actually put in about which pixel is next to what. You can scramble the rows, it's okay. So we didn't build in any spatial contiguity. Later stage, we might need that. And we're saying that we want to find a matrix D, we call dictionary, columns become the dictionary, and a coefficient A, matrix A, so that each of them can be written as a linear combination of these dictionary elements, and with positivity constraints. So positivity constraints, I start to appreciate the really powerfulness of positivity constraint. The first actually one was about lights. I was working on network tomography. And we did EM and everything, and eventually we end up with positivity constraint and some dynamic upgrading. That was enough. We threw away all the EM, which was very expensive. And if you think about in high dimensions, positivity constraint, this is, I don't know, hundreds and thousands dimensions put it together. It's the first quadrant. It's very, very small relative to the rest of the space. So with this positivity constraint, we didn't need L1 constraint. And you can see that the dictionary elements we find, we call principal patterns, are very spatially coherent. It's because I think the different genes respect these boundaries. So we have theory work I won't show here, shows that these different principal patterns shouldn't be on top of each other too much, right? Imagine if they're all on top of each other, there's no way you can linearly decompose, find them. But if they kind of zigzag, sometimes overlap here, sometimes over there, and relatively sparse, and you can recover them. So this contiguity without putting it in really shows to us that, yes, we find something. And we show it to the biologists, they were really, really pleased. They recognize the different shapes, roughly, right? It's not as precise exactly. The textbook decomposition building blocks are also schematic, not precise. And of course, non-negative matrix factorization, at least goes back to 70s, and there's recent surge of interest because of the Lee and Sung paper, and we use Morel's SPAM software. The same Morel with formula mentioned. Okay, so this is really a boring idea. It's that there are lots of ideas. Which one do you borrow? And later we discovered that non-negative metric factorization have been used in bioinformatics because we knew we didn't know that, but more as a way to do clustering, not this type of interpretable results for images. And the new contribution from my group is actually using stability principle to have a model selection criteria for how many of these principal patterns we want. I really see this sparse coding or non-negative matrix factorization based decomposition. You might want to do transformation first before you go to the linear regime. It's really, I see it more like modern PCA, because you really need to decompose. I mean, that's how we work best. We decompose the problem to simpler pieces. And PCA doesn't quite work, and we need to low dimensional structure. One way is find a linear regime and do sparsity. You might have to do transform, most time probably, but here seem to have worked. And the idea for selecting the number of components is kind of related to number of clusters. It's a very difficult, challenging problem. And for this, we think we find a good solution. So the idea is a stability, but how do you bring in the perturbation, right? We did have different labs. What we use is the random starting point. So this non-negative matrix factorization, in terms of DNA, is a non-convex problem. So you can have local minimum. And you know that for some simple case, we can plot it. The different local minimums actually are permutations of each other per D. Because what's principal one, it doesn't matter, right? So, but you want to match them. So how do you match them? We use correlation to match. We permute, find the best match. And then for two random starting points, we just do random selections from the observed images as the starting point. And then we end up with two dictionaries. We match them by permutation, use correlation, and this is the measure. And we minimize the instability. 21 was chosen, 22 is pretty good. And 22 actually decomposed one of the principal patterns in the 21 into two. And because we like simplicity, so we went for 21, okay? And this is a data set from one of the early papers on analysis of non-negative matrix factorization, Donahoe and Staden, almost 15 years ago. They had this cute swimmer data set. You have different parts of the body and you swim, and we find exactly. This is just, we have a mathematical proof of sparse coding, now for negative matrix factorization. So it shows you that, it works well when you know the truth. So give us some confidence that what do we find? Because that's why I suggested Siqi to work on the theories. My worry is that, and he solved the problem beautifully, that's the beautiful part, is that I worry that we found the wrong decompositions, and then you just screw up everything else. So these are the 21 principal patterns. I'm being trained mathematically. I couldn't help thinking this is the X and Y coordinate system, right? This is the vertical, and this is the horizontal. And there are other weird bits which I couldn't put into the X and Y coordinate system. And now you put it on this is called the fate map. And the reason you have these circles half green and half purple is because when we did the more precise mapping, this area fall into both. You have to rescale everything. So we think might be, and from the data, it seemed to be the case. So you now can give roughly classification or label of this different principal pattern. Remember, the principal pattern is just a region, and we think it's the pre-organ region. So at this stage, the morphology is now developed enough for humans to inspect and say, this is kind of tissue, this is the gut, this is the hind gut. So this is, but the biomolecules were already working, right, to build something. So this is something the old technology couldn't do, but we can. This is the pre-organ regions. And Then what, you say, right? Looks nice, kind of qualitative validation. So we believe now this principal patterns is a powerful computational lens to discover hidden structures in the linear domain. Why they're useful? So these are the things we're doing to provide evidence to say to us and to other people why this is a useful decomposition. I will show you that you can use these principal patterns to compare different genes, say which genes express where. So basically, think of these principal patterns as rooms. So this is an embryo, a big house with 21 rooms. Which team of workers go in there, do something? And especially among the 700 or so genes, about 20% of them are so-called computational genes. So these are the genes we know they bind somewhere, they're so-called genes, but we don't know their functionalities. So these give conjectures on what they might be doing, who they might be talking to do some job, and where do they appear. So that's one thing we can use for character. We have a website to put up for other people to explore. Remember the prediction, the supervised learning bit. All the previous work are done on supervised learning. Can we go back there, now use our unsupervised PP features and do the same prediction? How does it compare? And can we reconstruct a gap gene network, which I showed you, right? Use modern data. And the most important, the most, I think, solid proof of anything, can we go to the wet lab and do knockout experiments and then prove what do we find? And then can we use it for other data sets and discover hidden structures there? So we feel like we push ourselves pretty hard to ask the question, why this is useful? And we have a paper being revised for PNS. If you're interested, email me, I'll be happy to share. And we have 40-page support information, because one of the referees actually said that delete 2 sections because he thought it was not as well. It's still a great paper, but we felt if we delete the 2 sections, that we won't feel completely happy. So we did a little bit even more. So now it's even bigger SI.